My name is Robbie Fenster. I'm a visiting scholar in Miriam Heyman's lab at the Picower, and I work on Huntington's disease. It's a genetic neurodegenerative condition that is caused by a change in the Huntington gene. I see some patients who have Huntington's disease in my work. I've become very interested clinically in Huntington's disease, but it's also a disease that has it's a richly complex sort of set of questions, both from a scientific and sort of biopsychosocial way of thinking about things. It's a disease that runs in families, and with the genetic test that's now available, you can know at a very young age whether you're going to develop Huntington's. And so there's both a set of scientific questions that are quite interesting, as well as sort of both ethical and sociological questions that I find of interest. The work that I do at the Picower in Miriam's lab, or Dr. Heyman's lab, is that so this gene that's dysregulated or that malfunctions in Huntington's, it's expressed in all cells of the brain, but we don't really understand why it makes cells sick, and in fact it makes some cells more sick than other cells. And so in Dr. Heyman's lab, we're trying to understand what makes one cell different from another cell and why they react differently. And the hope is that if we could understand these differences, that we might be able to design some better treatments for the illness, because right now there are no treatments for Huntington's disease other than symptomatic treatment. Some of the work that we've done looking at these different cell types has identified some key players in the way that DNA is packaged and how that happens differently in the Huntington mice compared to wild-type mice, and that mutant Huntington interacts with some of these enzymes. We were interested to see whether there are any drugs that target this complex and whether they might have some sort of effect on Huntington's disease pathology, whether this could actually lead to a treatment. And so our next work is to try to understand whether if we reverse some of the changes that we see happening in the Huntington's mice, whether that makes the mice better. And then if that worked, going perhaps later on into people who have Huntington's.